These cats can glow in the dark. Now, is this a naturally occurring trait due to a random mutation? Nope, this is an example of genetic engineering, and we're going to take a look at how this process works in this video. Note that genetic engineering goes by different names. You might also hear it called gene splicing, recombinant DNA, or genetic modification. But all of these terms mean the same thing, which is basically this: taking DNA from one organism, combining it with another organism, and getting recombinant DNA, DNA from two different sources. Now, in the case of our glow-in-the-dark cat, most of the DNA came from the cat, but it received a gene from a jellyfish that allowed it to glow in the dark. Now, you might be wondering, how is it possible to combine DNA from a cat with DNA from a jellyfish? Those are two really different organisms. Well, the key is that the genetic code is universal. So if we were to take a look at the DNA of this animal, the DNA of this bacteria, and the DNA of this little boy, we would see some differences, but they're all made up of A, T, C, and G. Now, the sequence might be different, but they're speaking the same language. And because the genetic code is universal, we can combine DNA from different organisms, and genetic engineering is a reality. Genetic engineering relies on enzymes, two types in particular. So let's take a few minutes to see the role of these enzymes in genetic engineering. One of these enzymes kind of acts like scissors. It's known as a restriction enzyme, and its job is to cut DNA at particular sequences. And then there's another enzyme known as ligase, and its job is to glue DNA back together. Now let's explore restriction enzymes in a little bit more detail. There are many, many different restriction enzymes, thousands, and each enzyme is specific for a particular DNA sequence. So this restriction enzyme will only cut DNA when it sees the sequence CCTGG. This enzyme will only cut DNA when it sees the sequence TTCGAA. And this enzyme will only cut DNA when it sees the sequence CCTAGG. You get the idea. The other thing to note about restriction enzymes is that many of them cut unevenly. And this is known as the restriction site. So let's take a closer look at how the restriction enzyme cuts unevenly. Here's a DNA sequence. Here is the restriction site for a particular enzyme. It likes to start cutting here, but then finish cutting here. So what happens is that we get an uneven DNA sequence. These uneven DNA sequences are known as sticky ends. And they're sticky ends because since there's only one DNA sequence exposed here, it can easily bind through complementary base pairing rules to another sticky end. And this is important to genetic engineering. So here we've got our sticky ends. Now let's take a look at how we would use these two enzymes together to create recombinant DNA. So here's our DNA sequence. Here's a restriction site for a particular restriction enzyme. First thing we're going to do is add a restriction enzyme to cut it into fragments. There we go. And here you can see the sticky ends. Now, we're going to add DNA from another source that's also been cut using the same enzyme. And this is important because using the same enzyme means that you get the same sticky ends. So here's DNA from one organism, here's DNA from a different one, but same sticky ends. Now, these fragments are going to stick together because of complementary base pairing. So here we go. This sticky end was complementary to this sticky end, and this sticky end was complementary to this sticky end, so they naturally came together. However, we need a little ligase at this point because ligase will help attach the sugar phosphate backbones, which are more difficult bonds to make. So once ligase gets involved, here we go. We now have our recombinant DNA. DNA from one organism combined with DNA from another organism. So this is how we would put two different genes together. But at this point, pretty useless. All we have is a recombinant DNA molecule. In order for that to actually build a protein that makes a trait, we need to put this DNA into an organism. So now let's take a look at the whole genetic engineering protocol. And we're going to use a particular example, a very commonly used example of genetic engineering. And this example involves the use of bacteria to be human insulin factories. That's right, because of genetic engineering, we can make bacterial cells produce insulin that's not bacterial insulin, that's human insulin. And then we can bottle that insulin and give it to diabetics. So here's how it works. First of all, let's start with the bacterium. Why would we use a bacterium as a human insulin factory? Well, one reason has to do with its genetic structure. Bacteria have a big circular chromosome, but then they also have these smaller loops of DNA called plasmids. And it's very easy to move plasmids into and out of cells. So that's one advantage to using bacteria. The other advantage is that bacteria are unicellular and they reproduce asexually, which means that in a very short amount of time, you can get millions and millions of identical bacterial cells, clones. And this is important if you want factories. So let's see how the experiment works. We're going to start with our E. coli bacterium. Here you can see its chromosome. Here you can see its plasmid. And then we've got a human cell. And this human cell contains a nucleus, which contains DNA. And within that DNA is a gene for producing insulin. Now, this is the correct gene. This produces good insulin. So the first thing we're going to do is we're going to take the plasmid out of the bacteria, and we're going to take the DNA out of the human cell. Easy enough. Then we're going to cut both of them with the same restriction enzyme. So here the plasmid has been cut, and here the human DNA has been cut. And we're going to use a restriction enzyme that cuts at sites around the human insulin gene. So it's important to pick the right restriction enzyme for your experiment. So now we have our human insulin gene and we've got our cut plasmid. Now we're going to combine those two together. And because of complementary base pairing and because of the sticky ends, they're going to naturally bond together. Then we're going to throw in a little bit of ligase to help seal the sugar phosphate backbones. And now we have our recombinant DNA. Most of the plasmid is bacterial DNA, but it now has the human insulin gene. Pretty cool, but again, this is useless unless we put it inside a cell so that the DNA can be used to make the protein insulin. So let's do that next. Step number 7. We're going to stick this recombinant plasmid back into the bacterial cell. And this process is known as transformation. Whenever you give a cell new DNA, it's called transformation. So now we're going to let this bacteria cell reproduce. And in a matter of days, we'll have millions and millions and millions of identical bacteria, all of them with this recombinant plasmid that contains the gene for human insulin. So we're going to let these bacteria produce human insulin. And then what we're going to do is we're going to take the insulin out of the bacteria. And there's a special technique where we can isolate that protein. So now we've got the human insulin, and we can bottle this up, and we can use it to treat diabetics who need doses of insulin. And that, in a nutshell, is how to do genetic engineering. Keep in mind, though, that this is only one example. We can move genes from different organisms into other organisms. So one application is to make pesticide-resistant plants. These have a gene from bacteria that allows them to produce their own pesticide. We can make bacteria that can clean up toxic waste. We can produce proteins to dissolve blood clots. We can make growth hormone. We can even make acid-washed genes. And in class, we'll take a look at some other applications of genetic engineering.